This conference will now be recorded.
Okay, great. Well, we're almost at the top of the hour. So I'll wait until it turns to 3 o'clock, and there we go. So hello, everyone. Welcome to our live Q&A on COVID-19 with human milk experts. This discussion is brought to you by the Social Media Committee of the International Society for Human Milk and Lactation, also known as ISRHML. My name is Dr. Lauren Brink, and I will be your moderator today. I'm a scientist at RB, a global company that manufactures health, hygiene, and nutrition products. However, today I'm moderating as a scientist in the area of human milk, as a member of ISRHML's Social Media Committee, and as a former ISRHML Tigger, or trainee. As such, there will be no discussion of any RB brands or products. We are currently in the midst of a truly global pandemic, and today we wanted to spend some time discussing the research activities as well as the clinical implications of COVID-19. Joining us will be Dr. Shelley McGuire, Director and Professor at the University of Idaho, Dr. Lars Bode, Professor at the University of California, San Diego, and Dr. Paula Meier, Professor at Rush Medical College. For this hour-long conversation, we prepared a few questions in advance, but we invite the audience to submit questions in the chat box for us to potentially address at the end. And with that, I'd like to turn it over to our ISRHML members who have graciously given us the time to discuss this important topic. And so let's turn it over to you, Shelley. Could you describe your research interests related to human milk and what your involvement with ISRHML has been over the years? And I do ask anyone who's coming online if you could just turn off your videos, so it's just the four presenters on the call would be great. But yeah, let's turn it over to you, Dr. McGuire.
Lauren, thanks so much, and thanks for organizing this. I have been a member of ISRHML since 1986, so long-term member, and have been on the executive board and secretary treasurer for years, very involved in the society. Our research has spanned everything from milk lipids to pesticides in milk to most recently microbes in milk, and so this topic is sort of near and dear to our heart. I am a nutritionist by training, so.
Great, thank you very much, Shelley, appreciate it. And Dr. Bode, could you please introduce yourself, tell us about your research interests and involvement with ISRHML?
Sure, thank you so much for organizing the seminar in the first place, Lauren and Laura. So I'm a professor at UC San Diego and also director of MAMICOR, which stands for the Mother Milk Infant Center of Research Excellence down here in San Diego, which is funded by the Family Lars Rosenquist Foundation. I've been a member with ISRHML for about 11 years. 10 years ago, came on as an executive counselor. I've done that for 3 years and then was elected president in 2014, I believe. During my term, I started together with Sarah Mukarsel, the trainee interest group. So it's great to see that the trainee interest group, our dear Tiggers, are now taking on all these tasks. Thank you so much for keeping that alive. And certainly it is the most alive part of the society. Together with the Family Lars Rosenquist Foundation, we also started the trainee extension program. So that's where some of the connections are coming from. And tomorrow is going to be my last day as immediate past president. So I'm finally rotating off of the EC and will not be involved with this anymore. So last day tomorrow. Thanks for organizing this.
Yeah, of course. And again, if anyone's joining us, please make sure you turn off your video screens. And Dr. Meier, could you please introduce yourself, your research interests, and involvement with ISRHML?
Yes, I'm Paula Meier. I'm a professor of pediatrics and nursing at Rush University in Chicago. I've been an ISRHML member since 1997. I've also, like Lars, served as president and president-elect and past president, so I know what that feels like. It's 6 years worth of work. My research has always been in the area of human milk, lactation, and breastfeeding for premature and NICU babies. And since I'm towards the end of my research career, it's spanned so many areas. It's clinical, it's translational, all the way from health outcomes and cost of human milk for premature babies through to models of care delivery, which includes our Rush Mother's Milk Club peer counselor program. So clinical translational work.
Fabulous, thank you. So I'm going to just jump right into some of the questions that we prepared. And the first one's going to go to you, Dr. McGuire. What is the state of the science regarding the presence or absence of SARS-19 in milk? And what about other coronaviruses?
Great question. So we'll start with the current epidemic, COVID-19, and the virus that causes it, SARS-CoV-2. There are, to our knowledge, about 9 papers published looking at whether or not the virus can be identified in human milk. All but one of those studies was done in China. The other study was done in Singapore. They're all very small studies, and the good news, that's not me with the feedback, the good news is that none of them have identified or been able to find the virus in human milk. So that's positive. But being human milk researchers, we look at those studies and we say, but how was the milk sample collected, and was the assay validated, et cetera? And I think we still have a lot to learn about that. So even though there are a few studies out there, there's still a lot of work to do. You asked about other coronaviruses, other similar viruses. There have been a few studies done looking at SARS and MERS, which are similar coronaviruses. Again, there's been no virus identified in milk, but in those situations, we're talking about maybe 1 milk sample analyzed. Interestingly, for MERS, we do know that the virus shows up in camel milk. So if a camel has the illness, the virus shows up in the milk, and that's quite well documented. So we don't have a lot of data out there. And this is one reason why a lot of researchers right now are trying to figure this out together while the epidemic is going on, because you can imagine this is pretty difficult to study, and it really does have to be studied while women are infected. So I hope that answers your question.
Yeah, it definitely does. Thank you very much. Dr. Meier, I might ask you to maybe mute yourself. Yep, perfect. Just so that we can try and avoid that feedback. And the next question is going to be going to Dr. Bode. And let me see if I can get it back up. Let's see. And I wasn't able to print this out as well, and I cannot open it at the moment.
I can read it for you if you want.
Sure.
Okay, so question to myself, what are the immediate research needs in this realm, and why do we need experts in human milk and lactation to be engaged in this space? And I think Shelley set the stage here nicely for the questions and also for the urgency. We need to have a definite answer whether the virus is in human milk. That still does not set the stage for transmission through human milk. Just because it's there doesn't mean it gets transmitted. There's other questions that follow that. But if it's not there, the chances that it gets transmitted, of course, are very low. So I think that's an immediate question. And here it's important to know, yes, milk is a different matrix compared to all the other biological samples. And every assay is dependent on the matrix that, in this case, the virus is coming out. So it's going to be important to set up an assay that is validated for human milk. So in other words, you take human milk, you spike in the live virus, and you see how much can you recover. Do you get full recovery? Can you measure the virus actually that you put in? The next question is, and that really comes out of the human donor milk realm, what happens if we pasteurize the milk? So let's assume the virus is in human milk and we pasteurize those samples. Do we get rid of the virus? And if not, then that would be a huge problem for donor milk, of course. So 2 questions, one staged on the other. If it's not there, we don't have to worry about pasteurization. If it's there, then we need to make sure that pasteurization gets rid of it. I see that as the immediate and urgent field of questions. And then next would be an entire different question, and that is, what's the benefit of human milk? Can we identify any human milk components that potentially have antiviral component activity? Do we have antibodies that when mom is exposed, maybe even already during pregnancy, And then does she make antibodies, and do those antibodies go into human milk and protect the infant? Is there other components in human milk that potentially have antiviral activity? So it's really two different stages of research. One is immediate, and the other one certainly would help if we need.
Yeah, the second stage about the antibodies is making me think a little bit about, it reminds me of what's going on with the potential therapy that they're proposing, taking sera, people who've been infected, and utilizing the antibody as a treatment. So it is really interesting to see that. I mean, that is definitely probably going to be happening in human milk. So really interesting stuff. And and congratulations on the funding from the Willenhurst Foundation. Could you talk a little bit about how that project was initiated and how it's developing?
Right. So maybe to step up, I mentioned that during the introduction already, we have a strong relationship as ISRHML with the Family Lars and Rosenquist Foundation. We are teaming up with them, and they're funding the trainee expansion program. So that's the first step of interactions that we had. And then they're funding our research center, MamiCore, with an endowment. So we're really trying to connect other researchers at UC San Diego and really around the world to do biomedical research that is related to human milk. And really to tell you what an amazing partner they are, I sent an email to maybe someone needs to mute the screen. If you're not talking, please mute. There's a few people I see have their active microphone on. So just to tell you how amazing that partnership is, really, I sent an email at night to the managing director, Katrina Lichtner, at the foundation, and said, do you guys have any emergency funds? There is really an urgent need to get this research done right now. Anything you can do. And literally, I woke up in the next day, they're 9 hours ahead of us in Switzerland, and she came back and said, yes, we have a commitment, $100,000, we can do this, absolutely possible. And since we already had a relationship with our gift office and our advancement office, the process was extremely smooth, the money was wired immediately, and we have the money to get started. So this is really a process within 36, 72 hours, the funding was there. There is currently no time to write grant applications on this. There's no time for a lengthy review process or something like that. So it really matters that you have a relationship of trust, that you have an infrastructure that you can actually make those funds available. And since MamiCore was already working with FLRF before, it made it really a very smooth transition. So excellent partners, cannot highlight that enough.
I mean, it's really exciting that you guys have been able to get this going so rapidly. But there are a lot of people and a lot of different collaborators involved. And how do you all work together to ensure the research moves forward? And can you talk about some of the coordination that's involved in a big project like this?
Yeah, so it's really two different levels. One is on the local level, how MamiCore interacts with different labs here in San Diego, but also in other places of the world. And that's really what the center is set up to do. It's connecting the dots, finding people that work outside the space and bring their expertise into human milk research. So we've been working with the Center of AIDS Research, for example, here at San Diego, that have all the experience, that have BSL-3 labs, that can do all the different work that we need to do in this space. So it's really important to bring the experts, the virologists, the infectious disease experts to human milk, and not just the human milk experts to this specific project, right? We also have a milk bank in San Diego that's building right now. So there we have some expertise with Lisa Stellwagen leading that effort. And we have a human milk biorepository with an active IRB protocol that can sample and is sampling milk right now from North America, Canada, and US. So we can bring this all together and act fairly quickly. Now that's only at the local level. And at the same time, Shelley, Sharon Donovan, many other people, Kirsi, Aaron Seppo, Megan Azad involved, you really had a group of ISRHML scientists that got together independent of this. That said, let's do something, we need to work on this, and they organized a Slack group, so it's a team organization platform, and Shelley can talk about more about that, but it's a rapidly evolving platform as well, where you have multiple researchers from multiple different fields coming together and discussing ideas about protocols. How do we need to sample? What do we need to do to have the right assays up? So it's really multiple layers of people feeding into each other, and we're part of that as well. So it's really making resources available very quickly and knowledge available very quickly.
I'm really curious, Lars, about the logistics of this. I mean, we're all shut down, right? We're all working from home, and a lot of labs across the country are all shut down. I mean, are there exceptions made for this type of research, or what hurdles do you have to go through?
Right, so I'm assuming that depends on university or state or country you're in. The way it is, I can tell you for the UC system, we are on lockdown. So if you do not do any essential research, so assuming you have animal studies going on that if you would stop right now, you would lose months or years worth of work, you could still come in and finish that. But it's one person per lab, so you're not overlapping with multiple other people in the same space.
Yeah.
And research that is related to COVID-19 is allowed as well.
Okay.
So please, everyone else who's currently not working on any of this, stay home. That's the most important message I think that we have to convey to everyone over and over again. Please stay home. You don't have to work on this right now. All your milk samples can wait. I know the 10-year clock is ticking. I know there's other things going on, but please do not come to the lab right now, and don't send your postdocs or grad students. Stay home.
Shelley, do you have anything to add about working and logistics for lab during this time?
Yeah, I'll just echo what Lars just said. The same thing is happening at the University of Idaho and then neighboring Washington State University. So, you know, unless you're essential personnel, and again, it's the same definition. If you're working on COVID-19 or you're essential in some way or another, you really need to stay home. So we do have, you know, a couple people in the lab trying to move the assay forward, et cetera, but I'm pretty much working from home.
Yeah, and that's what we all should be doing, and then whenever we can. And we're quite lucky to be able to do so, if I do say so myself.
Maybe to add to that, I think many of us have these little manuscripts in the drawer somewhere that we need to finish up. And this is a good time now to pull them out, finish them up, submitting them. There's people that will actually review them because they're sitting bored at home. So you get those manuscripts out pretty quickly.
Yeah, the editors are going to be flooded soon, right?
I would like to mention one thing. Lars mentioned a Slack group. So this is a web-based platform. If you are interested in working on COVID-19 and breastfeeding, shoot one of us an email. And if you want to get involved in this Slack group, we can certainly add you.
Yeah, I think that kind of segues into our next question, was really, how can other researchers leverage the activities that you guys are doing such that everyone's aligned and insights are provided in a quick and rapid time? Is it really through the Slack group, or are there other avenues?
Well, I guess I'll just start on that. The Slack group is only one, and we're still reaching out to, you know, as Lars said, this needs to be an interdisciplinary an effort. I mean, we know about milk. We're not virologists, at least most of us aren't. And so the best way to handle this big problem is to get interdisciplinary groups working together. I can't overemphasize that.
So really utilizing that as a leverage point and a place where people can come together and bring different expertise and bring everyone to the conversation. So if you're interested, please reach out to Dr. McGuire. And you know, building off of that question too, are there other avenues of funding for researchers interested in the relationship between COVID-19 and breastfeeding?
Well, I'll just take a stab at that as well. If you have an NIH grant currently with an institute that gives supplementary administrative supplements, they are entertaining those sort of applications. And of course, there are a lot of emergency calls for proposals coming out right now. You know, I mean, this is an area that, as Lars mentioned, this is emergency funding. This is not normal sort of research funding that we're looking for right now.
Maybe to add to that, Lauren, there's also a chance to work with your advancement offices. If you have any donors that might be interested in this space, please reach out to your advancement offices. We have a GoFundMe page if you want through our advancement office, so everything matters. And if it is $20 or $50, if we all do that and we all reach out to our communities, that would also increase the money that we have available for this research. And that is much faster and much more uncomplicated than going through the funding routes right now, which will be important too. So don't disregard that. Do that. Some of those have deadlines with June 1st or July, and that's okay because we will be in this for a long period of time, and we need this research to be prepared for the next wave, which is coming either on this virus or when the next viruses are coming, which they will. So I think we are in a position right now to put protocols together, to put techniques together and technologies that next time we are better prepared than we were this time.
Great, thank you. I kind of would like to shift a little bit and talk a little bit more about the clinical implications of this virus. And so Dr. Meier, if you could, I mean, everyone, we've all seen, or hopefully we've all seen online, there's been a statement posted on the website about ISRHML and, or on the ISRHML website about COVID-19 and breastfeeding. And it does a great job of compiling the current studies that you mentioned, Dr. McGuire, and also the different recommendations from World Health, public health advisories. You know, reading through them, I just have to wonder, how realistic do you feel some of these guidelines are provided by various health organizations? And the one that really stuck out to me was the one from the CDC, where they recommend that if mother is too ill to breastfeed, that instead they pump. And I'm just finished breastfeeding my babies, and you know, pumping's challenging, especially if you're sick. So yeah, could you speak to that, Dr. Meier? How realistic are some of these guidelines for sick mothers and breastfeeding? And you're mute, so there you go.
Okay, yeah. So I've gotten so many personal emails apart from those with ISRHML, just people reaching out about the conflicting recommendations. And when you think about it, they're conflicting because there's really different avenues of thinking about the virus. And one of those is, since we don't have documented transmission into breast milk other than maybe your hands or the contamination, and that babies can be born without having been exposed in utero, the thinking is, okay, well then we need to separate the mother and baby, because at birth the baby would technically be symptom-free. And if the respiratory system is how the virus is spread, and not via breast milk, that is really the basis for one set of recommendations, which is isolate the mother and baby, except coming together for breastfeeding using a mask, or in some instances there are recommendations where mothers are actually separated from babies and they're supposed to pump milk and send it to someone who's unaffected to feed the babies. And legitimately, I think people raise the question of, certainly in some societies or even in some tight households, how realistic is it that there is going to be a person who is unaffected or unexposed to be able to feed the pumped milk to the baby? So I think that's one set. Is the baby's born without the virus, it may not transfer into the milk. We have no evidence right now that it does. So the transmission would be via droplet. Okay, so you separate. The other one is that I think if we look at the other recommendations coming out of World Health Organization, is that it may be impossible to, it's certainly in some countries, to have that degree of separation. So we rely on everything we know about previous viruses, and that the milk will protect the baby, that the baby should be in close contact with the mother. So I don't think it's unreasonable that we have different recommendations, because you have different schools about how to baby. But nonetheless, that doesn't help people in the clinical setting that really want to know best practices. As far as pumping, Lauren, I'm trying to understand your question. If your questioning, I think, is if mothers are very ill, is it realistic that they pump? Is that okay?
Well, yeah, I mean, breastfeeding can just be a lot easier, right? Because you can lie down, the baby can just be at the breast, but pumping, you actually have to sit yourself up. And so just I'm thinking about the mechanical logistics, and if you're really ill, is that a reasonable substitution?
Well, you can pump. I can tell you, having worked with infinite numbers of mothers that are very sick themselves, some in intensive care, we have certainly those mothers pump with babies in the NICU. So it certainly can be done. Is it easy, and is it necessarily the best thing to do? Again, it depends on which recommendation that you're looking at. And I think that the hard thing that people are struggling with is they want one answer, and we just don't have that answer right now. Because again, as Shelley and Lars have pointed out, the studies, we only have such few studies that show the virus isn't transmitted the milk. We have no studies that show if it is transmitted, how does the milk protect the baby? So I think everyone is struggling with what our best practice is with that clinically.
Yeah, thank you for speaking to that. And building off of just the impact of this virus in NICUs, how does this pandemic influence the donor milk supply? Could COVID-19, I mean, I know the question hasn't been answered whether this virus is in milk. It hasn't been detected yet, but you know, is there a chance that this is significantly impacting that supply from both just accessibility, there's not as much donors, to maybe it's compromising the supply?
You know, I've reached out and I haven't found anything on different web pages for donor milk use other than a call for more volunteers because of COVID and the need for milk donations. But Lars's point is well taken. We don't know about pasteurization. We would assume, I think I would, that if you've been through 2 freeze-thaw cycles and a pasteurization, which is most donor milk, and your only option is other than that of the NICU is formula feeding. I think that we have such documented risks of formula feeding for these babies. The best option would be mother's own milk. But I think probably if we really look at what are real risks versus theoretical risks on the NICU, we would have to come down to probably using the donor milk.
Okay.
If I may add to that, Lauren, I think the worst what's happening right now is the uncertainty. Uncertainty usually leads to giving wrong recommendations potentially, but really confusing moms, confusing recipients of that donor milk. And I would assume that that puts a dent into the supply. So the sooner we get answers to some of these questions that we raised earlier, and I think we're getting there within the next week or two, to really say that, yes, the virus is there, no, the virus is there, and if it's there, pasteurization either takes care of it or not, that will be absolutely critical. And I think it highlights the need for the research that's going on right now, because really recommendations from the major organizations depend on it. And here really the CDC, the WHO are looking to this group here for answers. So I would not disregard the importance of this. At the same time, I think ahead a few months, if you see that the numbers are now coming up on the African continent, for example, where the supply of human milk and the use of formula potentially has really life and death consequences, I really think that we need to have these answers as soon as possible. It really is absolutely urgent at this point.
Great.
I will add from the literature, for what it's worth, I know women aren't camels, but there's a large literature on the MERS virus in camel milk, because that was actually a public health problem where MERS was happening, and it's easily destroyed with pasteurization. So I'm hoping it's the same with this virus, but again, we won't know until we actually test it.
Yeah.
Right, and I would add, even with pasteurization, that's the final step, because you do have at least 1, if not 2 freeze thaws. So you've got that before the pasteurization. So I think that at least when I'm in a position clinically where I have to advise. So I think that makes it a little bit different. And I use the best evidence I can from, just like you were talking about the camels and the MERS, from other viruses and what we know, with the caveat that, you know, we really don't know, but I'm giving you, as a clinician, scientist, my best recommendation about this. And again, in NICU, I don't think in my mind, there's no question about the mother's milk in NICU, unless we were to find virus, and then I would still have to think, because the risk of formula for the babies that would be donor milk recipients is documented in terms of sepsis. It's documented in terms of necrotizing enterocolitis. So, and those are very real threats. So we have to, I think, balance what we know and what is theoretical.
Well, kind of building off of that, you know, we've talked a lot about what are the main research questions that need to be answered immediately and then secondary. Dr. Meier, could you talk a little bit more about what you're seeing as some of the real clinical challenges that clinicians are facing in the NICU related to the pandemic?
I don't know to the extent that we've actually, I know that we haven't seen NICU patients with COVID, but I'm sure that probably it's coming throughout the United States. One of the questions that I've been asked quite a bit is about the actual milk containers. So it really gets into many ways what you would think about if you were collecting milk for sampling and then sending it to some other, is how do you protect the people that are handling the milk? So I've gotten a lot of questions about, should we be doing anything about the exterior of the containers? You know, obviously, you know, I prepared just as a clinical perspective, an education sheet to use with families, because I think that families, for example, need to understand that the virus can get into their milk even if they don't transfer it through the breast, that they can handle their equipment, they can breathe in it, they can cough in it. And since NICU patients, the mothers are pump dependent, those are real important things to remind them. It doesn't have to come through the breast, it can come through your hands. But then the next question is, well, assuming that you have a mother who is positive for COVID, what about the exterior of the containers? So I'm getting questions, how long does the virus live on the milk collection containers and storage containers? Do they need a separate place in the freezer? Should they be, well, already, you know, those patients would be managed with gloves and, you know, full protective gear. So is it enough to receive them in the patient room with gloves and keep them in the patient room? But then what if they have to go to an overflow freezer? So there's a lot of questions. And if you're an institution that rents breast pumps, is there any special cleaning that's needed with respect to breast pumps? Can they be delivered into the neonatal intensive care unit from the outside just as they were before? So those are some of the questions that I think are again, they're not directly, is the virus in the milk, but they're trickle down questions that have a lot of clinical reality for people in the trenches here.
So, and then I know a lot of hospitals in the United States are baby friendly, such that room-in is like the clinical norm. With these with COVID-19 and the separation of the baby and mother potentially, are there additional considerations that are being done in that realm? Is rooming in still the way to go in these cases, or would babies potentially have to be separated?
I think that you'll see hospitals that are working both ways with this, but I do think that the trend is much more one of thinking about this, let's be on the safe side and separate the mother and the baby while they're in the hospital. And how much of that is really safe side? You know, you don't know, but it's the hospital's way, an infectious disease way of thinking, let's be on the safe side. We're not going to expose them in the hospital. But then again, you wonder if they go home in an environment where there's just the mother and the father in a one-bedroom apartment, how realistic is it that you're able to keep that distance and have somebody feed the milk that you've pumped? So I think there's a lot of what happens in the hospital is one thing, what's going to happen after the delivery and they go home is quite another.
Thank you. Yeah. So I'm wondering if this is might be a good time to open up see if there's any questions from the audience. And so if anyone wants to please submit questions in the chat, we can review them and have a discussion on that as well. But in the meantime, I wanted to have a discussion more about people who might not be as familiar with ISRHML. Could you guys speak to the group a little bit more as a whole, what the society is all about? Maybe Shelley?
Okay. What is the longest standing member here? The oldest, maybe? No. So yeah, the International Society for Human Milk and Lactation was established. It was started by a group of really excellent milk researchers, both on the dairy side and the human side. Really got started in the 1980s when people were realizing that, hey, guess what? Milk is hard to analyze, and human milk is an amazing food, and we need to understand what's in it. And so we're a society that made up of researchers and clinicians Very active society. We have incredible international meetings every other year and have for quite some time. And so if you are a student interested in human milk and lactation, if you're a researcher interested in human milk and lactation, we really would encourage you to look into the society. We're a relatively small society, pretty tight knit, and this is why we're able to galvanize everybody so quickly in this situation and other situations. And I'm sure Lars and Paula could add to that.
I'll pop in, Shelley, because I was actually, I think, one of the very first primarily translational researchers to be a part of the society. So one of the things that I enjoy the most about the society is that it is truly interdisciplinary. So people that do research in all aspects of human milk and lactation are a part of the group. So in a session, I remember one of my very first sessions at ISRHML, we took the HIV issue, and the people that presented in that session was all the way from the epidemiologists, the public health researchers, into the in the milk and the immune properties in the milk. So it was every aspect from the molecule to the population, I think was the name of one of our meetings. So it really is multidisciplinary, and I think there's also a unique appreciation among the researchers for the different types of work we do.
Yeah, I second and third everything that Shelley and Paula had said. One thing to add, maybe I mentioned it in the very beginning, I'm super excited about having the trainee interest group being very active right now. So it's really the young generation or the upcoming generation running the society really from the bottom up, which is amazing to see. So I highly congratulate you for putting this together, but really being this active group that that runs ISRHML really.
Thank you. Yeah, I know this presentation has been organized in part by myself and also by Laura Galante, a trainee down at the University of Auckland. So shout out to you, Laura, for helping out with this as well. And it looks like we do have a question from the audience. If mothers with COVID-19 using a breast pump, using pump for feeding, so I guess it goes back to the pump, a discussion around the pump's sterility. So how are they washed, and then also an access to pumps. Not everyone has access to pumps. So how do we marry? I mean, you have talked a little bit already about how proper handling of pump material needs to be determined, and that's a critical issue. But then also, what about access to pumps?
So yes, I think this is a critical issue, and that's why I think that the one recommendation, the idea that one is going to fit everything probably doesn't make sense, because there are certainly countries, especially some of our more resource poor countries, where pumps are just non-existent, and if they are, they're not necessarily effective. They can be easily contaminated with other types of viruses and bacteria that might even be more harmful to the baby than COVID. So yes, I think that is one of the legitimate criticisms of policies that recommend mothers are separated and use a breast pump, is that not only are they not widely available, but easy when you're using a breast pump to introduce other pathogens into the pump if it's not cared for properly. Now, it can be cared for properly. I know we teach mothers in NICU how to do that. But again, that requires, hot, soapy running water and things that other countries may not have readily available.
No, I think that's, it really, when I was reading through all the guidelines, man, it's just messy. There's just so many different guidelines. How is the mom to know? But, I think really putting it into context of what people have access to, and the geography in which the guidelines are provided makes a lot of sense. So thank you to speak with me.
Lauren, let me just add one more thing. And I think it's really important for people to understand that this is that ISRHML doesn't really do these guidelines.
Yeah.
We are basically trying to take these guidelines, putting them into documents for people to use. And what I've tried to do is to give what I think is the scientific background for the different guidelines. But that doesn't mean that we endorse one over the other, because nobody really knows right now. And believe me, as somebody who works clinically, I'm so empathetic with people on the front lines because they don't have the answers. We don't have the answers. And I wish we had more from ISRHML's point of view, but that's really not something I think we're capable as a group of.
Great, thank you. So we do have another question. I am interested in how our experts think that breastfeeding and general health in term infants will be affected due to the elimination of public health support because of social distancing, i.e. no more breastfeeding support groups, no prenatal care groups, even limited public health nurse visits to the home to weigh the baby and access breastfeeding technique. A lot of these support and social support groups are eliminated. So would you like to speak to that, Dr. Martin? Or Shelley? Or
I'll say one thing about that, because I think that this community within ISRHML is going to have other research questions rising out of this, other than is the virus in the milk?
yeah.
Because I think this is some example of it. You know, what happens with the social distancing? What is acceptable to mothers? I do think that especially some of our people doing more of the social science or behavioral work, and maybe even some of the economic work. But I do think that there are other questions about this that, again, are going to be a benefit if we revisit something like this again. But yes, the social distancing, even in the actual hospital setting, is a difficult one. So much less when you get out in the community.
I guess being a glass half full person, I would just challenge folks out there to figure out creative ways to work through this situation. It's amazing to see, even at the university level, how we're able to move everything to online in a matter of like 2 days. So maybe the silver lining here, we'll figure out ways to reach moms, maybe in a better way. I would hope that this might spur that.
Yeah, I mean, I think it's a good point that, maybe not everyone has access to Zoom or a computer, but I mean, everyone has their own smartphone, and there's definitely different online capabilities. It's really neat to see how, in this time of social distancing, I feel like it's bringing a lot of people together. So, like us right now. But so I have another question. Assuming the virus is not in milk and the infected mother pumps, what is the risk that her milk is contaminated post-pumping? So assuming the virus is not in milk, but is in the pump, what is the risk that it's contaminated post-pumping? And I think, Paula, you've addressed this a couple times already, but it does seem that, we're worried, people are worried about contamination within the pumping chain of activities.
It's interesting because what the researchers think about, I would say the collection of the milk samples that you think about for research is really very similar to what we think about and how we handle these things clinically. So it's very, very similar in terms of looking at if the virus isn't in the milk, we want to make certain that it's not put there some other way, both because we're going to study the milk, but also because we're going to feed it to the baby. So I think that analogy is really clear. And the best thing with that, again, is going to be the proper hand washing, making certain that the kit is appropriately cleansed. And there are all sorts of guidelines about how to do that. as well as not coughing, sneezing, or breathing on the kit while you use it. And that actually is common NICU practice for mothers who are breast pump dependent. Is common NICU practice because anything can get into the milk in the process of the pumping it and storing it if you're not or in handling it in the NICU. So anything can contaminate it along the way, and that's why you have to be so careful. Both what you would do for research to be sure you've got the correct sample, you've got to do clinically to ensure it's appropriate for the baby.
Maybe one thing to add that we haven't talked about yet, and I think that's important both for research but also for evaluating the risk, and that's this peculiar virus-specific age tropism that we see, that it's mostly the elderly. That's not 100% true anymore, but usually you see in pathogen situation that it's the elderly and then the most vulnerable infants. In this case, we don't really see that too much. So yes, we have a few cases there as infants or children, and a few preterm infants that were contaminated through during pregnancy already. But we don't really see this usual spread that we have the very young and the very old being most susceptible. So maybe in this case, we're buying ourselves a little bit of time here, but for the next virus, that might look different again. So again, I can only highlight this again, be ready for the next pandemic that will likely come. This is not a surprise to scientists at all that we have this pandemic right now. It's a question of readiness in the end as well. So these questions are not just for this specific virus. This is a question. These are questions that we need to answer faster during the next pandemic.
Yeah, well said. And I mean, I think it's really interesting that it's not just the question of contamination is not just in the clinic, it's also in the research setting, right? We're human, we interact with samples in the lab, and we all have to maintain the best PPE in all instances. So I think that was a nice point. And we have another question.
Maybe to that, Lauren, please, that's a very important point as well. There's a reason why there is experts that work with viruses and BSL-3, BSL-3+. So while we're trying to set up BSL-2 and BSL-3 labs ourselves right now, there is certain protocols and certain training that if you rush through this, you expose your personnel potentially to a very high risk. So please don't just start something in the rush of something. Give it to the experts that are already working in BSL-2 or BSL-3. And don't start something just because you think this is the hot topic right now. We need to do this. We're milk researchers. We can do the virology as well. Let's set up a BSL-2. So please don't.
Okay, I think it sounds like the feedback is gone. So, but thanks for that comment, Lars. So yes, another question from the audience. Scientists are working on isolating SARS-CoV-2 antibodies from serum obtained from patients, recovered COVID-19 patients, to be used as a treatment. Any thoughts on human milk as a source of antibodies?
I think we talked about that earlier already during the introductions, what the research questions are. That certainly is an interesting question. Mom is exposed maybe during pregnancy, maybe during lactation. Maybe she doesn't even know. There's no tests, but maybe those antibodies pop up fairly quickly. We have no idea what's the case there. So that will require research where you take time course samples and see when the virus-specific antibodies pop up. And there is labs that have assays set up for this right now. So that's certainly something that is happening as well.
Has that been done on any other pathogens in the past, looking at time course of infection to antibodies in milk?
I believe so. I'm not sure exactly if I have an example for it right now. Shelley, you've done some work with the Gates Foundation there. You have any insights when that pops up usually?
Well, it's my understanding it's usually about a week later. But again, I'm no expert on that. One thing I would like to add, though, is I think there's also an interest in perhaps the infant coming in contact with the virus and having the mother respond to that via breast antibodies. So again, it's a question we don't know the answer to, but like Lars said, this will take time course sampling, which is very possible to do.
Thank you.
And I had another component.
There's a lot of feedback.
Everyone, mute themselves, that would be great. I think Shelley, you're now you're. There's other milk components as well that might respond to the virus. We've seen this with HIV, for example, and oligosaccharides, that we see moms that are infected produce different oligosaccharides. We also see that moms that transmit make different oligosaccharides. So both transmission risk as well as infection is, in the case of HIV, somehow related to oligosaccharide. And there might be many other milk components that change when mom is infected, and that might be potentially beneficial to the infant or do other things. So I think there's a lot of research that needs to be done, maybe not urgently, because that's not urgent questions right now, but certainly something to follow up on. And we're all collecting samples, and we'll be able to analyze those and answer those questions.
I think that's really interesting, right? It's not just the IgA antibodies, but the potentially HMOs or other components that might be responding to these viral infections. I think that's
Lauren, if I could add, I think the other thing we want to think about is, one thing is what's in the milk, and the other thing is how does it interact with the baby's saliva, the stomach acids and all. Because some of the other viruses we know that the gastric environment is very hostile ordinarily in a human milk-fed baby, and it takes very little formula to change that pH. So I think the whole idea of exclusivity, to me, in relation to other viruses, if we're going to breastfeed, it should be as exclusive as possible, just to maintain that hostile gastric pH. So I think there are just little things from other viruses when we think about mechanisms, same as coming in contact with the saliva of the infant. So I think there are other mechanisms that are not just the milk, but how they interact with the recipient infant that probably are worthwhile to think about.
That's great, thank you. So here's another question. How do you recommend, so someone's diagnosed with COVID-19, a lactating mother, and wants to support science, how would you recommend to save samples from patients for further research, future research?
I think I'll take that one for now. And if anybody hasn't muted, please mute because there's some feedback. So the Slack group that we talked about last week, about a dozen of us put our heads together and came up with interim guidelines on how to collect the human milk sample for COVID-19 research. These guidelines may change over time, but it was we used the knowledge that we collectively had to come up with those guidelines, and they are available on the ISRHML website for anybody that is interested in collecting milk for this purpose. And again, they might change over time, but that's our best educated guess right now as to how to collect and save those samples for future research.
Thank you. Do you think that a mother who's diagnosed, a lactating mother who's diagnosed with COVID-19, do you think it can be their treatments for COVID-19 in particular need to be changed? Are there any additional implications or precautions that physicians need to be on the lookout for lactating mothers and the treatment of COVID? That's one of the questions that we received. I'm getting up.
Yeah, I have no idea. It's something I hadn't even thought about, so I don't know if Shelley or Lars would.
So, I mean, I guess, if I'm thinking about that question, for lactating mothers, drug nutrient interactions, drugs get into the milk supply and affect the human milk, and so we're talking about these anti-malarial drugs that Trump has been promoting, at least. Are there any literature on those drugs and how it might be getting into the human milk?
Let me take that, maybe, if the feedback is okay here, the feedback that we get. I think that is general questions. What if mom is now put on medication because of the virus? But that's really the case for any other disease as well. If mom is put on medication, we don't have a lot of information whether that medication makes it into milk into the infant. There's a lot of research needs to be done at that. Christina Chambers at UCSD, that's the main reason why we started that milk biorepository, is really to look at medications in human milk. And in this case, it's a lot of medications that are currently tested that are repurposed, right? So that's medications that are meant to be for other diseases and that people are screening right now in high throughput to see if we find an already approved medication that can be used against this virus as well. So it would be great to have some of that information. For some of the medications, we do have it. Very often you read on the little leaflet there that comes, if you're breastfeeding or pregnant, ask your physician, and the physician will tell you, I have no clue, so you better not breastfeed. And that's, of course, is, again, a completely wrong recommendation because of lack of data. So again, it's this research community that can make some of those, close some of those research gaps that lead us to giving wrong recommendations.
Any other ads, Dr. McGuire? Maybe this is a good time. We have about 5 minutes left, and I know the feedback's quite bad, but any final words or comments from maybe Shelley, we can start with you.
I'll just add that, you know, we're all in this because we understand how amazing human milk is as a living fluid, living tissue. And there's so many things to learn about it, not just the nutrients that are in it, the cells and the viruses and what should be there and what shouldn't be there and how they respond. And there's really no other that's like it. So this is just one of a million things to study about human milk.
Thank you. Dr. Bode?
I'm not sure if I have too much to add to that. I think for everyone who's currently not in that mix and currently not working on coronaviruses in human milk, doesn't mean that your research on human milk isn't important. So please keep doing what you're doing, even though if you have to sit on the couch for a month right now and you can't go into your lab, there's other things you can do. Please keep doing what you're doing because human milk is indeed powerful and we have so many unanswered questions. So not everyone has to jump on coronavirus right now. Your work still remains important for the next 10, 20, whatever years. So thank you for everyone who's doing that and who's involved in this field. You know, especially again, I can only highlight this, the trainees, you're the future of this field, so much appreciated.
Okay, thank you. Dr. Meier, any parting words?
Yes, I would just remind everyone that regardless of the fact that some of the actual recommendations are in conflict, what isn't in conflict anywhere is the recommendation to continue breastfeeding. So that's across everybody. That's World Health Organization, CDC, Academy of Breastfeeding Medicine, is to continue breastfeeding. And I would say in the NICU, especially, it's probably even more important with babies having their own mother's milk. But and I think a lot of the actual recommendations will work themselves out according to the hospital, the culture, the access to pumps. But the main thing is we need to support the idea that breastfeeding is still best, and that for me is the most important thing.
Well, thank you all very much for your time. I've really enjoyed our conversations, and I do hope you all stay well in this trying time.
Thank you, everyone. Thanks for organizing.
Thank you.
Stay well, everybody.
Thanks a lot.
Can I ask, it's Megan, ask our speakers to smile because I'm going to take a screen.
Oh, yeah, we could do that. Let's see.
Go back. Okay, good. On three. 1, 2, 3. Great. Thanks.
Bye. Thank you.